Good morning. I am Madhurima Datta, Zoology and Biology Educational Content Creators. Now content for B.Sc Zoology Honors, M.Sc Zoology, CUT PG and CSI and Life Sciences student. Paper non-chordates or invertebrate zoology, unit Platyhelminthes, and topic is general characteristics of blood fluke and life cycle of blood fluke. Blood flukes or schistosoma. Introduction. Schistosomasis is an infection with blood flukes of genus Schistosoma. Schistos means divided and zoma means body. Ranks among the major infectious diseases in the world with 200 million people infected. Epidemic. The disease is widely prevalent over much of Africa and parts of South America, West Indies, Middle East, and Far East. The old generic name for the worms was Bilharzia, from the Theodor Bilharz, that is German parasitologist who discovered Schistosoma hematobium, and the infection was called bilharziasis, a name still used in many areas. Description. Blood flukes differ from most other flukes in being dioecious and having the two branches of the digestive tubes united into a single tube in the posterior part of the body. Males are broader and heavier and have a large ventral groove. The gynecophoric canal, that is the posterior to the ventral sucker. The gynecophoric canal embraces the long, slender female. Three species account for most schistosomiasis in humans. Schistosomiasis manosi, which lives primarily in veins draining the large intestine, and schistosomiasis japanosium, which occurs mostly in veins of the small intestine, and schistosomiasis hematobium, which lives in the veins of the urinary bladder. Next, intermediate host. Freshwater snails, including species of Bulinas, is the intermediate host. Definitive host, human, that is the most important definitive host. Other mammals such as cattle, dogs, and rodents can as the reservoirs. Life cycles of blood fluke. At first, the egg stage. Based on the species of worm, female fluke measuring up to 25 millimeter in length, releasing approximately 3,500 eggs daily into the bloodstream. These eggs migrate to the intestine or bladder and are released into the environment through feces or urine. Next, the larval stages. First, miracidium. When the eggs are in the water and under favorable conditions, they hatch the released miracidium larva. Miracidium larva is the ciliated larva that swims to the intermediate host in the case of snails. Next, Cerceria larva. After further development, the larva transform into a fork-tailed larva called Cerceria. The Cerceria larva emerge from the snails into the water. When these larvae get in contact with a mammal, they drop their tail and penetrate the skin tissues where they feed on blood. Once the Cerceria larva drops its tail, they become schistosomula. Next, schistosomula. The schistosomula travels through the lungs to the heart and later enter the liver where they undergo development. After the schistosomula becomes mature, they exit their liver through the portal vein system. Adult stage. During this stage, the male and female worm are sexually mature. The male and female worms mate. The adult worm reside in the intestine and this location varies depending on the species. They do not remain in one place and migrate to different locations inside human beings. The eggs produced by fertilized female worm are moved to the lumen of the intestine, the bladder, the uterus, and released via feces and urine. Then the entire cycle begins all over again. Next, schistosome dermatitis, that is swimmer's itch. Various species of schistosomes in several genera cause a rash or dermatitis when their cerceria penetrates hosts that are unsuitable for further development. Cerceria of several genera whose normal hosts are North American birds causes dermatitis in bathers in northern lakes. Severity of the rash increases with an increasing number of contacts with the organism or sensitization. After penetration, Sarsaria are attacked and killed by the host's immunomechanism, and they release allergic substances causing itching. The condition is more an annoyance than a serious threat to health, but there may be economic losses to vacation trade around infested lakes. Next, Paragonimus or lung flukes. Several species of Paragonimus, a fluke that lives in the lungs of its host, are known from a variety of mammals. Paragonimus is a western money from East Asia and the Southwest. Pacific parasitogens, a number of wild carnivorous, human, pigs, and rodents. Its eggs are coughed up the sputum and swallowed, then eliminated with the feces. Zygotes develop in the water and the miracidium penetrates a snail host. Within the snail, miracidia give rise to sporocysts, which in turn develop into radial larvae. Sarsaria larvae form in radia and are then shed into the water or ingested directly by the freshwater crabs. They prey on infected snails. Metacersaria develops in the crabs, and the infection is acquired by eating raw or undercooked crab meat. The infection causes respiratory symptoms with breathing difficulties and chronic cough. Fatal cases are common. A closely related species, that is Calicotti, occurs in mink and similar animals in North America, but only one human case has been recorded. Its metacersaria live in crayfish. Now topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima for playlist of B.Sc Zoology Honours, M.Sc and CSI Net Life Sciences, and also biology classes for class 9 to 12. Thank you.